So again, the goal of translation is to make the protein, and in order to make the protein, what the cell reads directly in order to do that is this messenger RNA molecule. You might remember that proteins are made up of a bunch of amino acids that are just strung together. And so another way of thinking about translation is that it's a process where a cell uses the information on a messenger RNA in order to figure out what sequence of amino acids should get strung together in order to make a protein. It turns out that when a cell reads a messenger RNA molecule, the way it reads it is it reads it 3 nucleotides at a time. So it actually reads each set of 3 adjacent nucleotides, and it's each one of these sets, so it's each set of 3 nucleotides that encodes for a particular type of amino acid. So again, that's how basically a cell reads that messenger RNA. It's each set of 3 that encodes for one particular type of amino acids. These set of 3 nucleotides that encode for a particular amino acid, they're called codons. Right? So again, in words, codons they're found on messenger RNAs, and what they do is that they encode for a particular type of amino acid. And there's 3 nucleotides found in one codon. So that's generally how a cell uses the information on messenger RNA in order to make a protein. What we'll do next is we'll show you more of the details. But before we do that, how about if we mention that someone has actually figured out about 65 years ago exactly which amino acid each one of these codons encode for. So if you think about messenger RNA, there are 4 types of nucleotides found within it, right? There's A's, U's, G's, and C's. Codons are made up of 3 adjacent nucleotides. So if you think of how many different ways can you string together 3 letters, if there are a total of 4, turns out that there are 64 possible ways to do that. And again, someone has figured out what amino acid each one of the 64 codons encodes for. And so that information is contained within this thing right here. It's the genetic code. And again, every single codon that's out there, every 3-letter combinations of U's and A's and C's are listed within this table. And also listed within this table is exactly what amino acid each codon encodes for. So for example, if you're interested in finding out what the codon AAU encodes for, you just look that up in this table, and it says here that AAU, whenever the cell sees that on a messenger RNA, that encodes for the amino acid asparagine. You can see by looking at this table that each amino acid can be encoded for by more than one codon. So for example, the amino acid leucine, that will be kind of inputted into the messenger RNA whenever the cell sees any one of these 4 different types of codons. And so we'll see the significance of that starting next week. So why is it significant that multiple codons can encode actually for the same amino acid? One thing we'll point out about this table now, though, is this. So notice that one of the codons, AUG, it's considered to be a start codon. And that's because basically translation begins whenever the cell runs across this AUG codon. So another way of saying that is that if you consider all of the proteins that a cell makes, AUG encodes for methionine. AUG also happens to be the start codon. So the very first amino acid found in all of the proteins that a cell makes initially, the very first amino acid is methionine. It turns out that cells eventually can take off that methionine that's at the start and replace it with something else. But when a protein is first built, all proteins, when they're all first built inside of a cell, they all have the amino acid methionine at the beginning. And again, the reason why is because the codon that encodes for it, AUG, also happens to signal where translation should begin. Right? That's why it's called the start codon. Along the same lines, if there's a start codon, you might deduce that there's also stop codons. And it turns out that there are 3 different stop codons. So whenever a cell runs across any of these, UAA, UAG, or UGA, those all tell the cell this is where the protein ends. So this is where you should stop doing translation. But again, the bottom line here is that someone has figured out what each of the 64 codons encodes for in terms of what amino acid it encodes for. So now we'll take a look at more of the details as far as how translation happens. How is a messenger RNA read in order to make a protein? So here are all of the things that are required in order for translation to happen. And so I kind of think of translation like baking, right? So whenever you bake like cookies or something, you need certain things in order to bake cookies. And so those same basic things are needed in order for a cell to make a protein. So if you think about what all is needed in order to bake cookies, you need a kitchen in order to do the baking. You need a recipe. You need someone to go get the ingredients, right, to make the cookies. So you need someone to go to the grocery store, get all the eggs and the flour and the sugar in order to bake the cookies. You need the ingredients themselves. It might be useful to have a timer, right? So if you're making many batches of cookies, it might be useful to have a timer to let you know when the baking time is up. So when you should take the cookies out of the oven and put the new batch in. And also, it might be helpful if you're going to maybe bribe someone to go to the grocery store to get the recipes and bring them back to the kitchen. It might be helpful to give them some food, right? Because that's kind of tiring to keep going to the grocery store and pick up stuff. So it would be useful to have some food for that person so that they don't stop doing their job. Right? So all of those things are needed in order to bake cookies. Those same types of things are needed in order for a cell to make a protein. So again, here are all the things that are needed in order to make a protein. And so what we'll do next is identify what all of these things are and generally tell you what they all do. So this here represents the messenger RNA. And again, the messenger RNA was made during transcription. And what the messenger RNA does, it's kind of like the recipe for making cookies, except that what it's the recipe for here is the recipe that tells a cell how to make a particular type of protein. Right? So you need instructions, you need the recipe in order to make a protein. Another thing that's needed in order to make a protein, well, just like in baking a cookie, you need the actual place to do it. Right? You need a kitchen in order to bake the cookies. So same thing when proteins are being made. You need the actual physical place in order to make it. And that physical place is the ribosome. So the ribosome is kind of like the kitchen in that it's the place where the proteins are actually made. One thing to mention about ribosomes is that if you consider each individual ribosome, it's actually made up of two parts. It's made up of a big part and a small part. And so, in other words, it's made up of a large subunit and a small subunit. If you're considering what is a ribosome made of, so what's the stuff that makes up the ribosomes in your cell? It turns out that ribosomes are made up of a mixture of proteins and RNA. It's made up of a type of RNA called ribosomal RNA, or rRNA. And again, the reason why it's called ribosomal RNA is because it's RNA that's it's RNA that's found in ribosomes. And so because you know that ribosomes are partially made of RNA, then you might deduce that there are actually nucleotide sequences that are kind of embedded within the ribosome, right? Because the ribosome is made partially of RNA, and here's the RNA sequence. And so we'll show you why this is significant in a second, that ribosomes are made partially of RNA. All right, there's other stuff that's needed in order to make a protein. So just like making a cookie, you can't make cookies if there are no ingredients. And so in order to make a protein, you need the ingredients. And the ingredients needed to make a protein are amino acids. So here's one example of an amino acid called methionine. And again, the function of amino acids during translation is that it serves as the ingredients. In other words, it's what proteins are made of. That's what you string together in order to make a protein. We mentioned that when you're making cookies, you need somebody to go to the grocery store and get the ingredients and then bring them back to the kitchen so that they can be used. Same thing is needed when a cell is making a protein. So somebody or some molecule needs to grab the ingredients and bring them to the kitchen so that they can be used. The molecule that does that during translation is represented by this thing. It's called a transfer RNA. So again, it's like the grocery store runner. It's the thing that grabs the ingredients or the amino acids and then brings them to the kitchen, or the ribosome. One thing to note about transfer RNA, so this is yet another type of RNA. So we've mentioned that there is messenger RNA, right? And we've shown you the job of messenger RNA here. There's ribosomal RNA, which is the type of RNA that's found in ribosomes. And now we're saying that there is a third type of RNA called transfer RNA. And in a second, we'll show you why it makes sense that these things are called transfer RNA. But for now, what we'll mention is this. So if you remember, during the first week of the quarter, we said that RNA can sometimes fold up upon itself in order to form secondary structures. And so here's proof of that. So transfer RNA, basically it's a single-stranded RNA that has become folded upon itself. Here's another picture of a transfer RNA to kind of hammer home that point. It's a single-stranded RNA, but it's kind of found some complementary sequences within the same strand so that they can form hydrogen bonds. And because those hydrogen bonds are forming, it gives transfer RNA kind of a T-looking shape. Right? So that's its secondary structure. And so because transfer RNA is a type of RNA, it has RNA sequences on it. And so that's what this is showing. We'll show you in a second why this is significant, that transfer RNA is made of RNA. Other things that are needed in order to make a protein, there's a couple of more players here, and here's what they do. We mentioned that when you're talking about making cookies, the person going to the grocery store to get the ingredients, it would help if you feed that person. Otherwise, he'd get tired and he'd stop going to the grocery store for you. So you need food for the grocery store runner to give him energy. The thing in the cell that serves as the energy molecule is ATP. Right? So the role of ATP during translation is that it provides energy for the transfer RNA to go grab the amino acids. And then lastly, we mentioned that when you're baking cookies, it would help to have a timer to let you know when to take the cookies out of the oven. Same thing goes when you're making a protein. It'll help to know, or it'll help to have something to help end the process. And that's what this thing represents. It's called the release factor, and it's kind of like the timer. Right? It helps the process end. So here are all the things that are needed in order to make a protein during translation. Here's what they all do in a nutshell. And so what we'll show you next is all of these guys in action. All right, so how do these molecules work together in order to make a protein? Here we go. So this is what all this shows is the cytoplasm of a cell. So if it's a healthy cell, it should have some free-floating amino acids in there that it's acquired through metabolism. And it should have, if it's a healthy cell, a bunch of free-floating transfer RNAs and release factors as well. We mentioned that the thing that's needed in order to make a protein, one of the things that's really needed, is the instruction booklet. Right? The messenger RNA carries the instructions on how to make the protein. It turns out that in order for translation to happen, the ribosome first has to bind to this messenger RNA. And it turns out that there's a sequence on the messenger RNA that kind of directs the ribosome where to bind. So we'll highlight that sequence here. So the sequence that tells the ribosome where to bind, it's called the ribosome binding site. And that's where actually the small subunit of the ribosome first binds. And the reason why the small subunit can bind there is because, remember we said that the ribosome itself is partially made of RNA. So there's an RNA sequence on the ribosome that's complementary to the ribosome binding site sequence on the messenger RNA. So because they're complementary, hydrogen bonds can form, and that's what allows the small subunit of the ribosome to attach to the messenger RNA. So in words, here we'll jot down what the ribosome binding site does. It's a sequence found on the messenger RNA that tells the ribosome where to bind to the messenger RNA. All right, we mentioned, so this is important, right? This sequence here, highlighted in yellow, tells the ribosome, the small subunit of the ribosome, where to first bind. Another sequence that plays a big role in beginning the process of translation is the start codon, and so here it is, AUG. And so basically what the start codon does is that it tells a cell where to actually begin constructing the protein. Right, so it's at the start codon where the first amino acid is brought into the process. So one of these amino acids is going to be brought to the start codon. The thing that brings the amino acid to the start codon is a transfer RNA. So it turns out that there's a transfer RNA that works specifically with each particular codon. The one that works with this codon is the transfer RNA that has the complementary sequence. Right, so this transfer RNA, UAC is complementary to AUG. So that tells you that this is a transfer RNA that's going to end up bringing an amino acid to this start codon. And so that's what happens. UAC, it turns out, interacts with the amino acid methionine, and it brings it then to the ribosome and the messenger RNA. And again, the reason why this transfer RNA can bind to this codon is because it has a complementary sequence. UAC is complementary to AUG. Once this first transfer RNA has brought the first amino acid to the messenger RNA, what happens next is that the rest of the ribosome, the large subunit of the ribosome, ends up clamping down on the whole process. And so now we're kind of seeing why ribosomes are needed during translation. Basically, the job of the ribosome is that it helps stabilize the interaction between this transfer RNA and this messenger RNA. Right? So it makes sure that the amino acid truly does get to the codon on the messenger RNA. And so what happens next is basically the same thing, right? So whatever the CCA codon encodes for, there's going to be a transfer RNA that brings a specific amino acid to it. So now we have the beginnings of a protein. We have the first two amino acids now strung together. You might remember that the bond that holds together two amino acids is called a peptide bond. And you might also remember that most bonds require some kind of enzyme to catalyze its formation. You remember that most enzymes are made up of proteins. Some enzymes, though, are made up of RNA. And so the enzyme that's catalyzing this formation of this peptide bond here, it's actually the RNA found within the ribosome that's helping to form this peptide bond between these amino acids. Right, so again, now we have the beginnings of whatever protein is going to be made. We have our first two in place. And so again, the same thing just happens over and over again. So whatever this AUU encodes for, there's a transfer RNA that's going to bring the amino acid there. But in order for that to happen, the ribosome has to kind of ratchet over, has to move over in order to accommodate the incoming transfer RNA. Right, so now once it's moved over, whatever transfer RNA interacts with this AUU, it ends up bringing its amino acid there. Once it's there, the RNA within the ribosome will help form this peptide bond now between that new amino acid and the rest of the amino acids. And we're really on our way now to making a protein. At this point, we'll mention this. So it turns out that you see where each of these three transfer RNAs are now kind of resting up against the ribosome. The place on the ribosome where they rest are given certain designations. And here are the designations. So there's an E site on the ribosome. E stands for exit. There's a P site on the ribosome. P stands for peptide. And there is an A site on the ribosome. A stands for acceptor site. And so here's why each one of these sites is given its name. So it turns out that this transfer RNA that you see here, it's already done its job, right? It's already brought its amino acid to the kitchen. And so after it's done its job, it can then leave and maybe grab more amino acids. And so that's exactly what it does. After it's already done its job, it ends up leaving, goes back. And so you see why it makes sense that this portion of the ribosome is called the exit site. So we'll rewind it. It's because that's where the transfer RNA, after it's done its job, that's where the transfer RNA ends up exiting or leaving. The reason why this is called the peptide site is like this. So how about if we show you what happens next? So whatever UAC encodes for, there's a transfer RNA that'll bring an amino acid to it, but before that happens, the ribosome has to ratchet over one in order to accommodate it. Alright, so now we can show you the reason why this is called the peptide site is because the transfer RNA that's sitting at that site, that's a transfer RNA that's holding the peptide that's on its way to being made. Right, so it's kind of holding the thing again that's on its way to being made. The reason why this portion of the ribosome is called the acceptor site, or A, is because this is the place on the ribosome where the new incoming transfer RNAs dock. Right? So whatever whatever UAC encodes for in terms of amino acids, there's a transfer RNA that'll bring that amino acid there. And so again, it's appropriate that this is called the acceptor site because that's the portion of the ribosome where the transfer RNA comes and brings its amino acid. Right. So basically the same thing happens for all of these codons. The ribosome ratchets over. Right. So after this transfer RNA has done its job, it leaves. That allows the ribosome to ratchet over. And so whatever UCC encodes for, there's a transfer RNA that's going to bring its amino acid there. A peptide bond ends up forming. This transfer RNA has done its job, so it can leave. And then the ribosome ratchets over. So this all occurs over and over again until the ribosome reaches a stop codon. So UGA is one of the stop codons. And so what occurs whenever a stop codon is encountered is that instead of a transfer RNA bringing an amino acid, what happens instead at a stop codon, so to show you that this is a stop codon, we'll highlight it in red or reddish. What happens at a stop codon is that a release factor binds there. And after the release factor binds, that kind of helps pry apart this peptide, this protein, from everything else. And so the protein ends up getting released, and then eventually everything else ends up coming apart. And so kind of again at the end, the big picture, what's the goal of translation? It's to make a protein. And so here it is. Here's a string of amino acids that was made by using the information that's contained within this messenger RNA molecule. And so translation basically at this point is finished, because there's the protein and there's the messenger RNA that was used to make it. So what we'll do last here as far as translation is I just mention a few miscellaneous things. Right? So again, each one of these groups of 3 nucleotides that's found on the messenger RNA that tells the cell which amino acid to encode for, those are called codons. We mentioned that each transfer RNA that brings an amino acid to the messenger RNA, at its base here is also a set of 3 nucleotides, and basically what it does is that it helps the transfer RNA bind to the messenger RNA. Right? So whenever there are complementary nucleotides on the base of a transfer RNA, that'll help it bind to, again, the complementary codon. And so the name given then to the set of 3 nucleotides at the base of a transfer RNA that helps the transfer RNA bind to a particular codon, because it's the complement, it's called the anticodon. So anticodons are found on transfer RNAs. And again, in words, what the function of the anticodon is, it's a 3 nucleotide sequence found on transfer RNA that's complementary to one of the codons that's found on messenger RNA. Again, we showed you that this release factor does something. We kind of said that it's analogous to a timer during baking. But to be more specific, what it does during translation is that it binds to a stop codon whenever one is encountered. And after it does so, it ends up releasing the peptide. So again, basically this is it. So a transcription makes a messenger RNA from DNA, and then during translation, here's where the messenger RNA is used to make a protein. All right, so big picture. In order to make a protein, DNA is used to make messenger RNA. That then goes to the ribosome in order to make the protein during translation. It turns out, though, that the proteins inside of cells, they aren't just a straight string of amino acids. You might remember that they end up getting folded and modified before they can be a fully functioning protein. And so what we'll do next is we'll show you what all happens to a string of amino acids before it can be considered a fully functional protein.